A lot of effort has been put into researching psychiatric disorders, but there is still no cure. It could be because we don't have a complete picture of the exact molecular mechanisms. Structural biology is one of the best ways to understand how small molecule drugs interact with the target protein. I'm Azadeh Shahsavar. I'm a structural biologist at the research lab of Paul Nissen at DANDRITE, which is the Danish node of Nordic EMBL partnership. In this project, we wanted to study the glycine transporter GlyT1 that sits at the cell membrane in the brain and modulates neuronal activity. Over the past 2 decades, many research groups have tried to find a way to inhibit GlyT1 and this way to treat psychiatric disorders, but even the most advanced inhibitors have failed in late stages of clinical trial studies. And we wanted to understand why. We aim to determine the 3-dimensional structure of GlyT1 to be able to see and understand how inhibitors interact with the protein.
I'm Gleb Burenkov. I'm coming originally from Russia and I'm a beamline scientist at EMBL Hamburg. I'm working very close with users from all over Europe, helping them conducting their research. EMBL Hamburg provides rather unique service, and what we are offering ranges from sample preparation and characterization through the X-ray experiments and data acquisition to data analysis and interpretation.
Crystallization of membrane proteins in general, and GlyT1 in particular, is not a straightforward task. EMBL Hamburg was absolutely critical in crystallizing GlyT1, and collecting data was only possible at the powerful, bright microfocus P14 beamline.
We actually achieved such a power of the X-ray beam that the experiment on single sample that takes roughly 1 minute, we managed to scale it down to a fraction of a second.
But EMBL is more than just excellent science and excellent infrastructure. It's also its people, their passion for science and their dedication to enable research at its best. When we collected diffraction data from several hundreds of GlyT1 microcrystals, the available software wasn't successful in analyzing them. Scientists at EMBL Hamburg developed a script that could successfully analyze such data. Once we determined the 3D structure of GlyT1, we were actually surprised because we didn't expect the inhibitor to bind the protein where it's binding. I believe it will form the basis for further pharmaceutical efforts to find different strategies targeting this protein in the brain.